How are you? कैसे हैं आप लोग? What are you able to see in that? What are you able to see on the slide? You're able to see different type of what is that? Yeah, what is it resembling like? Something that you daily cook in your household, in your kitchens. What your mommy uses all these particular things. How do they appear like? Well, they are the seeds. So today's session, which we are following from the earlier sessions which you have taken regarding endosperm embryo, and today's session, children, well, it is the seeds, right? Well, this is the next, I think, the product fertilized, post fertilized product. Well, this is the called as fertilized ovule, the final product of sexual reproduction. Well, children, next, what are the learning objectives? To define the seeds and perisperm, to observe and identify the parts of the seeds, to compare and interpret dicot and monocot seeds, to analyze the different types of seeds, to draw the sketches of both types of seeds, to critically think about its benefits for human purpose. What may be the purpose of the seeds, right? Okay, next one. A typical seed consists of seed coat. So when you see a seed, when you just break the seed also, it has a seed outer part, it has a seed coat. It may have one or two cotyledons and an embryonic axis inside it. Then the outer layer of the seed coat, that is called testa. See, just remember, the seed is actually the fertilized product, right? Fertilized product of what? Of the ovule, okay, which is enclosed inside the ovary. So ovule, you have seen that. If you just recall when you're seeing the structure of the ovule, different types of ovule when we discussed in earlier sessions in our video, anatropous ovule, orthotropous ovules. So ovule consisting of two types of walls. What do you call that name as? Integuments. That integuments only are still present in the seed, because the ovule what is becoming into the seed, isn't it? So the ovules names what is assigned now here. So the outer integument in the case of seed is called testa. Please learn like that. Okay, children. Next, the inner layer of the seed coat is called tegmen, and micropyle is there in the ovule. You know that, you would have already known the function. That is a small opening, small pore like opening through which the pollen tube enters, right? So that micropyle still persists in the seed coat, so through which all the that's where the seeds, when you soak in the water, why it bludges, it just swells up. That is a one physical force, the physical phenomenon that is called as imbibition. So how the water and all these things are entering through that, through the small pore, this is what is micropore, right? Next, the cotyledons of the embryo are thick and fleshy. Okay, children. Next, structure of the dicotyledon. Now we see about the dicotyledon seed. So earlier we saw about the dicot embryo. You just remember the diagram. Now you see that how this has become in the seed. Inside the seed coat is the embryo. So you can see this embryo. The parts, what is that? Plumule, hypocotyle, and radicle. And plumule actually give rise to the shoot axis, and radicle give rise to the root axis. Right. Right. Now that is the embryonic axis, and the two cotyledons you can see, they are very fleshy and they're full of reserve food materials, right, children? Now at the two ends of the embryonic axis are present the radicle and the plumule. So you can see here, and there is a hilum is present here, and there is an opening of micropyle through which all this hilum also you should remember, right? How this ovule is attached to this walls of the ovary, right? You remember placenta to which the particular there the funicle is present, and then hilum is also present, right? So that hilum is what this region is, right, children? So this is about the dicotyledon seed. Next, now about the monocotyledon seed. So how the monocotyledon seed is present? In monocotyledons, seeds are endospermic. So they have endospermic, and some, as in orchids, are non-endospermic. Something like orchids, the endosperms are non-endospermic. That we will see in the next part of the same session. We will try to see what is again endospermic seeds and non-endospermic seeds. In the seeds of cereals such as maize, the seed coat is membranous and generally fused with the fruit ball. So Even the this we will learn. Next part is called fruit. The fruit also they have layers, right? That we will discuss. The endosperm is fully and stores food, so it is full of nutrients. Okay, the outer covering of the endosperm separates the embryo by proteinaceous layer called aileron layer. The aileron layer is the outermost part of the this seed. Okay, this outer covering of the, we can say, outer covering of the endosperm, you have to say. It's not of the seed. Okay, of the endosperm. So of the endosperm, so this is the endosperm here, which is the nutritive tissue. So outer covering of this endosperm, no, they have a proteinaceous layer called aileron layer. So this is only seen in monocotyledons. We don't see aileron layer in the case of dicotyledons, right? So some differences are, you should note down. So that is a proteinaceous covering of the endosperm in the monocotyledons seed. Okay, children, you are seeing the parts of here. Now what's the scutellum here? You recollect from the previous session, that is scutellum is nothing but the name of the single cotyledon. Okay, and you can see all the axis is present here. That's the scutellum, and cleorrhiza and cleoptile. That is the cleoptile is the outer covering of the plumule, and sheath covering the plumule. Cleorrhiza is the covering of the radicle, right? So these points, you should keep it in remember by illustrations. You have to draw the diagrams and label the parts by which it will try to imprint in your brain. Okay, next. Now this is the comparison. So you can comparison between the dicot seed and the monocot seeds. So if you compare here, you can see where the endosperm is located here, and then where is the epicotyle, hypocotyle, and radicle, say radicle, and this is cotyledon. So we have two cotyledons, single cotyledon, that's called scutellum, and seed coat. Okay, and the difference that is in the endosperm, I said you that what is the outer layer of this endosperm in monocots is called aileron layer. Very good. Next. Now here, that's I said, whether that seed is based on its conception of this endosperm. So based on that, the seeds again are classified into two types: albuminous seeds and non-albuminous seeds. So see, albuminous seeds or endospermous seeds. So endosperm is not completely used by the developing embryo. See, the endosperm is the nutritive food which is actually required for the development of the embryo. Embryo is actually the seedling, future seedling actually, right? So they develop into the future, and there's a seedling once during the germination. So for that, for the development of all these things, endosperm. So the endosperm, the embryo is completely developed, but still the endosperm is not exhausted or consumed. Such type of seeds are called as endospermous seeds. So examples, so we have already seen the coconut, castor, maize. These are all the examples of albuminous seeds or endospermous seeds. Non-albuminous seeds or non-endospermous seeds. Endosperm is completely used up, so no endosperm will be there. So embryo is completely grown up and they is completely developed by the time the endosperm is completely exhausted or completely consumed. So it's completely used by the developing embryo before the maturation of the seed. The seed is before its maturation, it doesn't have the endosperm at all. Now, so there is no endosperm left in the seed. So the examples of this is pea, bean, and mustard. Is it here? Right. Next one interesting one is called perisperm. So what's called perisperm is nothing but a layer of the nutritive tissue. In the earlier slides we already seen what is the learning objective. So you have to define what is a seed as well as a perisperm also. So perisperm is something a new topic you are learning in this for your purpose, for your study purpose. So it is a layer of nutritive tissue in the seed of certain flowering plants that are derived from nucellus. So just recall what is nucellus. It is a nutritive tissue. It is a pre-fertilization product. It is a pre-fertilization substance that's present inside the ovule. It's present outer to the embryo sac, enclosed within the integuments. So that nucellus is actually meant for the development of the female gametophyte. But in some plants, even the nucellus will remain. Just remember like that. So in seeds of some plants, remnants of, it's a spelling mistake here, children. Nucellus, not nucleus there, is persistent, is still persistent. So the residual persistent nucellus is called perisperm. So you can give the example of black pepper. Such thing is called perisperm. Okay. Next. We have some assignment for questions for you. First question is, what is the outer covering of the endosperm in monocot seed? Second question is, describe the neat diagram of dicot and label parts, hilum and radicle. Next one, differentiate between dicot and monocot seeds with suitable examples. A dicot albuminous seed, this is MCQ question. Which among this is a dicot albuminous seed? Pea, castor, bean and gram. Next, a dicot X albuminous seed among this, a pea, castor, bean and gram. So please answer all these questions and please submit it. Okay, right. Okay, hello. Hey, hello. Yeah, yeah. Thank you and stay healthy. Stay safe. Bye. Read well. Gain your knowledge. Bye.